Thanks for logging in, and thanks to Molecular Devices for giving me the opportunity to talk about our experience with Ironworks Barracuda, more specifically about some of the experimental approaches for voltage-gated ion channels we developed within the last few years. In essence, I will argue that sub-gigasil automated patch platform Ironworks Barracuda is suitable for testing compounds on modulation of voltage-gated ion channels held in controlled conformation and at steady state. There's approximately 300 genes encoding ion channels. Each gene may have hundreds of SNPs and mutations. Some of them are functionally relevant, and around 100 have been associated with channelopathies. In addition, ion channels can heteroligomerize and form complexes with associated proteins, which can in turn serve as drug targets. The complexity of potential drug-ion channel interaction is enormous. Historically, ion channels present a fruitful class of drug targets. This slide lists a few commonly prescribed drugs targeting ion channels. It is noteworthy that most of them are older drugs, and nearly all were discovered empirically. Our understanding of ion channels is continuously improving and opening the novel possibilities for rational drug discovery approaches. For example, genetic mutations such, for example, CYP or erythromyalgia point to NaV1.7 as a target for pain therapy. Snail toxin-derived peptide ziconotide clinically validates anti-calcium channel as a target for pain. The target-based ion channel drug discovery efforts were, however, so far not very successful, and this is reflected in a few new approved drugs targeting ion channels and lack of return on investment, and then shift of research and discovery investment away from ion channel research. In some cases, whole therapeutic areas with substantial amount of ion channel targets get deprioritized. While the industry struggles with bringing new ion channel drugs to market, the scientific advances in our understanding of ion channel structure and function advance rapidly. This is to a large degree due to technological breakthroughs, such as the ability to solve crystal structure for ion channels. The example on the left shows, this is from Genentech work, shows the binding of aryl sulfonamide to the binding pocket on the voltage sensor in domain 4 of chimeric NaV1.7. To correctly interpret structure-activity relationship and guide compound optimization, functional data are needed as well. The selectivity profile for this aryl sulfonamide is shown on the right. The data were obtained with manual patch recording, where special care was devoted to maintaining the sodium channel isotype in equivalent conformational state. The complexity of ion channel drug interaction and the need for physiological relevance require assays with substantial throughput and good control of ion channel conformation to assure predictive value for the downstream in vivo endpoint. Electrophysiology, with its ability to control the membrane potential with its direct readout and sub-millisecond temporal resolution, is often the most suitable technique. The low throughput of manual patch is, however, not adequate for support of industrial drug discovery, and automated electrophysiology was introduced within the last 15 years to address this need. In a broad sense, automated electrophysiology instruments can be divided into gigasil instruments capable of high-fidelity voltage clamp and sub-gigasil instruments with advantage of higher success rate. This talk will focus on Ironworks Barracuda, which was introduced 5 years ago and follows Ironworks HT and Ironworks Quattro in the line of sub-gigasil instruments. The main objective of Ironworks Barracuda was to retain the functionality of Ironworks Quattro, primarily the high success rate and stability of recording. A schematic of one of PPC plate wells is on the left, and briefly, at the bottom of each well, there are 64 holes which get partially or completely occupied by cells on cell addition. Then amphotericin or similar perforating agents is perfused from the bottom, establishing electrical access. And then electrical circuit composed of common ground and individual recording electrode clamps the membrane potential and record the current. Recordings at nearly 100% success rate have become a routine, and screening in electrophysiology became feasible. The limitation of Quattro is in its discontinuous voltage clamp, which is a consequence of separate liquidic and electronic head. Barracuda aims, in addition to meeting the capability of Quattro, to overcome these limitations by integrating fluidics and electrophysiological recording as illustrated in the center. The silver one is the electrode and the bluish is the pipette. This integration enables continuous clamping of memory potential and recording of current during the compound addition. Complex voltage clamp protocols and recording of ligand-gated channels became feasible on Ironworks Barracuda. An additional advantage of Ironworks Barracuda is in its parallel 384-well recording compared to sequential 48-channel recording on Quattro, which gives it higher throughput. In our hands, Ironworks Quattro was very stable, robust, and excellent for screening purpose. But due to the lack of continuous voltage clamp and its reliance on pulse-based protocols, Ironworks Quattro assays had some limitation in supporting the structure-activity relationship, in particular with accurate potency determination of the inactivated state of the channel. And this was reflected in disconnects between Ironworks Quattro and Patch Express generated IC50 values for a set of screen-active compounds as illustrated in this graph. In our experience, Patch Express was a better predictor of potency in manual patch and also efficacy in animal models downstream. So we reasoned that the expanded functionality of Ironworks Barracuda may be suitable for the continuous voltage clamp-based protocols such as the ones used on Patch Express in manual patch. In this way, it would be a better predictor of downstream assays while retaining the high throughput of Ironworks Quattro. This is a look at the NaV17 currents recorded on Ironworks Barracuda in population patch mode. Depolarizing step evokes a current which rapidly activates and inactivates, as shown in A. The distribution of currents was relatively narrow, as shown in B. The average amplitude in our hands was around 2.5 nanoamp. C and D show activation of the current with a threshold at minus 50 millivolts and peak around minus 20. And E and F show inactivation with a half inactivation voltage of minus 50 millivolts. Overall, the properties of NaV17 current recorded on Ironworks Barracuda looked comparable to the properties of NaV17 currents recorded in manual patch. To accurately evaluate the effect of compounds on defined conformational state of the channel, we needed to assure that the biophysical properties of the channel remained stable throughout the experimental protocol. For that reason, we monitored channel activation and inactivation at the beginning and at the end of 30-minute recordings. In our initial experiments, we used cesium fluoride-based internal solution and observed large shift in channel activation and inactivation during the experiment. Later on, we switched to potassium gluconate-based internal solution and added series of wash steps and did dramatically improved stability during individual run and also during the 6 runs of typical experimental day. The bar graph down at the bottom show at the beginning and at the end for both activation and inactivation kinetics. Next, we designed a protocol capable of assessing the effect of compounds on channel in rested and partially inactivated state using multiple protocol modes of Ironworks Barracuda. Holding potential was initially set to a voltage where most channels will reside in resting state with protocol 1, and current amplitudes in response to 20 millisecond depolarizing steps are plotted in B at the bottom. Next, once the current was stabilized, we stepped the memory potential to a voltage corresponding to approximately 40% inactivation. This is the protocol called T3 at the top. And test compounds were added during this step. Finally, we returned to the memory potential to the resting membrane potential, and that is the protocol step 4. Block, the partially inactivating potential, was measured during protocol 3, and resting state block was determined by comparing currents evoked during protocol 1 and protocol 4. 2 additional quality control protocols, protocol 2 and 5, were added to monitor NAV17 activation inactivation. Experiments performed in population patch mode, patch clamp mode of Ironworks Barracuda are associated with substantial leak current conductance, uncompensated access resistance, and a non-uniform electrical field. Therefore, it was important to evaluate the uniformity of inactivation from well to well and between experiments. The example in this slide illustrates the distribution of inactivation across a typical patch plate. The frequency distribution of inactivation was narrow, as shown in histogram in A, and the spatial distribution of inactivation across the patch plate was uniform, as shown as a heat map in B. We were able to routinely obtain recordings with an average fractional inactivation of approximately 50% and uniform distribution of inactivation. The key strength of our protocol was its inability to evaluate compound effects at steady state for 2 different channel conformations. In this example, 1 micromolar tetracaine nearly completely blocked NAV17 currents at the polarized membrane potential and had almost no effect on the channel at the hyperpolarized membrane potential. From CRCs, we can see that there was approximately 100-fold shift in potency between resting and inactivated state. A similar evaluation was performed for several different sodium channel blockers, and this table compares the data from Ironworks Barracuda and data generated by manual patch electrophysiology. In general, there was a good agreement between the 2 methods for both compound potency and for state dependency. The Ironworks Barracuda I just described was highly reproducible, as illustrated by retest of 70 compounds. IC50s were obtained at partially inactivating voltage, and the correlation coefficient was 0.96. We further assessed the key advantage of determining a block at a fixed fractional activation is the ability to directly compare potency of compounds across multiple sodium channel subtypes that may inactivate with different kinetics and across different voltage ranges. A set of sodium channel inhibitors was tested for block of 6 sodium channel subtypes. Each subtype was depolarized to a holding potential corresponding to 40 to 50% inactivation. Most sodium channel inhibitors on this list display similar potency for all channel subtypes, suggesting that fractional occupancy of resting and inactivated state was indeed similar across subtypes. An exception is sulfonamide, which was selective for TTX-sensitive sodium channel subtypes over TTX-resistant NAV15. Our Ironworks Barracuda also provided useful information about the kinetics of compound association and dissociation from the target. Figure A and B illustrate the fast kinetics of association and dissociation for lidocaine and relatively slow kinetic for sulfonamide. Summary data for a set of 8 compounds is in the table below. In conclusion, it is likely the development of sodium channel modulator with superior therapeutic window will require good selectivity, strong state dependence, and an optimal kinetic profile. The assays I just described can support a drug discovery program by providing information on all these 3 attributes at a throughput required for industrial drug discovery project. So we next examined the design of a similar concatenated protocol to measure block of calcium current and encountered some unexpected difficulties. This slide illustrates barium current conducted by a recombinant N-type calcium channel and recorded on Ironworks Barracuda in population patch clamp mode. Similar to what is seen in manual electrophysiology recordings, the current activated rapidly and inactivated slightly during the depolariting pulse. The current voltage relationship shows a relatively steep voltage dependence of current activation, most likely resulting from imperfect voltage control on Ironworks Barracuda. However, the stability over time was excellent. Steady state inactivation was examined with a series of 1-second conditioning pulses, followed by a test pulse to 0 millivolt, as shown in B. Fitting the Boltzmann equation to the test pulse, amplitudes indicated a half inactivation voltage of approximately minus 20 millivolts. Similar to the voltage dependence of activation, steady state inactivation was stable in time. The protocol for assessment of compound effect on channels in resting and inactivated state was similar to the concatenated protocol I just described for sodium channels, so I will not go in detail. Similar to the sodium channel protocol, this protocol had the ability to determine compound effect on channels in resting and inactivated states within the same recording. Example in B shows that 1 micromolar trox had little effect on current evoked from hyperpolarized membrane potential, but almost completely blocked currents evoked from the more depolarized membrane potential. In CRC curves, we can see that there was approximately 100-fold shift in potency. Although initial experiments looking at activation and steady state inactivation curves indicated good stability of the recording over time, we wanted to assess the stability of currents at partially inactivated voltages in more detail. To this end, we calculated the block produced by addition of buffer during the depolarized holding potential. Heat map of the fractional current shows 4 areas of patch plate where currents were substantially reduced. The areas of reduced currents coincide with the location of 4 ground electrodes situated underneath the patch plate. And the impact of such non-uniformity on concentration response curves for trox is illustrated in B. Considering that silver chloride is a major component of ground electrodes, and also based on previous reports of T-type calcium channel blocked by silver ions, we hypothesized that the non-uniformity seen in our recordings may be caused by silver ions coming off the ground electrode. Therefore, we evaluated the effect of 2 silver salts on block of N-type calcium currents. The block by silver chloride is shown at the top, in the top row A and B, and the block by nitrate at the bottom, C and D. Both salts block N-type calcium currents with IC50 of approximately 0.5 micromolar, and the block was not state dependent. We next explored the potential for chelation of silver by DMPS, a heavy metal chelator approved for clinical use in cases of heavy metal poisoning. It has high affinity for silver and relatively low affinity for calcium and magnesium. At high concentration, DMPS applied in the extracellular solution blocked N-type calcium currents with approximately 5 millimolar IC50. However, addition of 1 millimolar DMPS to the buffer completely prevented block of calcium currents by silver nitrate without significant effects on N-type calcium current themselves. We next tested the effect of DMPS on the stability of N-type calcium currents in Ironworks Barracuda concatenated protocol. Addition of DMPS to our solution reversed the loss of current over the ground electrodes as shown in the bar graph. It was the most effective when DMPS was added to both internal and external solution. DMPS prevented the loss of current during the buffer addition and resulted in uniform distribution of current calcium currents shown in the heat map of the fractional current. To examine potential effect of DMPS on the pharmacology of N-type calcium currents, we titrated a set of well-characterized calcium channel blockers in the presence of DMPS. IC50 values obtained on Ironworks Barracuda in the presence of DMPS generally agreed well with the published data suggesting that DMPS does not alter channel pharmacology. In summary, the voltage control of Ironworks Barracuda seems to be sufficient for evaluation of compound action on controlled conformation of ion channels at steady state. The limitations of Ironworks Barracuda remain in sub-gigaohm seal, lack of signal conditioning, limited liquid handling capability, and in some cases toxicity of components of recording apparatus. In a broad sense, the incremental advances in automated electrophysiology continue to expand our ability to investigate ion channel drug targets in great detail and increase throughput. Before I end, I just want to acknowledge that all this work has been performed with support of Eli Lilly and Company. Mark did most of the Ironworks Barracuda recording, Mike all of the manual patch, and Birgit was the key to wrap up this project. We also had the access to general expertise in the Ironworks Barracuda, in particularly Yuri Kurishev from Chantes, Dave Rock from Essen, and several colleagues from Molecular Devices, Jim Konstantin, Xin Yang, and in particular Ed Perdon. So I appreciate your time and interest, and I'm passing the control over to David.